of the fountain of the water of life, that overcomest shall inherit all things, and I will be his God, and he shall be my son. And he said unto me, it is done. I am Alpha and Omega, the beginning and the end. I will give unto him that is thirsty of the fountain of the water of life. Anybody else?
Chris.
Psalms 27. Proverbs 27. Just a sec. Oh, Psalms 27. Father, forsake me, and the Lord will take me up. Wait on the Lord, be of good courage. He shall strengthen thy heart. Oh, wait, I say, upon the Lord, the is my light.
Nicole. Oh.
Thank you for singing.
Yes, thank you for leading, and thank everyone else for singing. What beautiful things we have to sing, God's Word. It's been a blessing this weekend, Creation Week all week, and then this weekend, all these different aspects of understanding things better about the whole debate of evolution and creation, and to see that we certainly can see the facts that are there, the data, and see it pointing to a gracious God who created it all. Let's begin now in prayer. Dear Father in heaven, I just pray again for your blessing in our presentations this evening, this afternoon. I pray for your Holy Spirit to come and help us to understand the things that are being presented, to be able to rightly divide the facts in the first book of yours, that of nature. And I pray that you would bless, that we would definitely understand and be able to have useful things to share with others later to help them see that you are our Creator and we can trust your word. I pray this in Jesus' name. Amen.
Okay, this afternoon we're going to get more into the content, the evidence relating to evolution. And the things I'm going to talk about, you mostly will not read, you'll not hear about on TV, you won't read it in the evolution textbooks. But if you read the primary research literature and understand what's really going on, then you find out things are different from what are in the textbooks. And it's not just that you have to be a creation of the modern synthesis. And that's really evolution as it's understood today. Now, keep in mind, even at this point, molecular biology was still in the future. They really didn't know anything about molecular biology. I guess we're getting some feedback, maybe from these microphones, I don't know. Anyway, so molecular biology really got going since the 1950s. In the last decade or two, it's just growing exponentially. I mean, remarkable growth in understanding how to study DNA and lots of other things. There? Okay, is that better? Okay. So there's been a rapid change in knowledge since Darwin's day. And this can't help but have an influence on what we know about how animals can change. So what do we know now that we didn't know a few decades ago? Some years ago, I was giving a talk to a group of teachers, school teachers, and one of them asked me, why are evolutionists becoming so much more confident of their theory? Is there new evidence that is causing that confidence? Okay, I'm going to talk about that today. There's the recent developments are rapid and spectacular advance in genetics and molecular biology, understanding what the cell, how the cell works. And that's something you have to know to understand how and if evolution happens. Okay, a couple definitions just to make sure we know we're talking about the same thing. The word evolution is tricky because it means so many different things. If we make a statement like we don't believe in evolution, to many scientists, it'll sound like we're saying a lot more than we really are. Microevolution is different from macroevolution. Microevolution is adaptation, changes within a species to allow animals to adapt to new conditions. Those two lizards are the same species, Euta stansburiana. The one on the left lives on the gray granite rocks around in Southern California. The one on the bottom lives on red sandstone in northeastern Arizona. They've each adapted to their environment. Okay, and it's clear God made organisms with the genetic ability to adapt within the range of potential that He put within them. And so that's very real. That's called evolution. In fact, most of what evolutionary biologists study is microevolution. And if you read people who, the writings of biologists who are criticizing creation, if you read carefully, most of the arguments they're bringing for evolution are microevolution and speciation, the development of new species, and that all is very real. It happens within the creatures that God has made. Macroevolution, that's where we go different directions. Did mammals really evolve from reptiles? Did we evolve from primates? Those kind of big changes, that's the place where we go different directions. Macroevolution, evolution of new major groups of organisms, orders, classes, phyla, which means you have to evolve new things like skeletons and kidneys and human brains and violinists and all the rest. Okay, so that's what we're talking about today. So microevolution is compatible with creation. It is adaptation since the creation. Macroevolution is contrary to biblical creation. So how does microevolution work? What is the process? The accepted theory is any change begins with random mutations, changes in the DNA, random mutations, and then natural selection. Okay, why random? Well, okay, these two concepts are the core of Neo-Darwinian evolution. If there's no creator, how are you going to make something new? If there's no creator, nothing could know what is going to be good for the animal. And this is a very basic concept in Darwinian thinking. There can be no process in nature that can see ahead and see what the animal needs. If there's no intelligence involved, the only changes you can make are random changes. Okay, and those are mostly destructive, and it's natural selection which has to weed out those that are not good. So those two ideas there, random mutations and natural selection, is the core of Darwinian theory. The process must be random. Nowhere in the process can there be foresight of what would be beneficial to the organism. Because if there's any indication that there's foresight, oh, this will be good for the animal, then there's intelligence monkeying with the system somewhere. And that's not allowed in Darwinian thinking. Only natural selection, which is a naturalistic process, determines what random change will succeed. Natural selection is very simple. If you and your buddy are out in boats, you're in a speedboat, your buddy's in a rowboat, and there's some monster there that's hungry. Okay, which one of you is going to survive to produce offspring? Well, that's natural selection. There's nothing complicated about natural selection. It happens. Faster rabbits will outrun more coyotes. And so in evolutionary theory, that's the only process that can determine what's going to be good or what isn't. You cannot have any foresight, knowing what will be good for the animal. Foresight would imply creation or intelligent design. And I have a little analogy here of evolution. So just look at the part on the right. Pretend this is in an automobile factory. You have a machine that makes pistons. And on the top of it, there's a computer that controls the machine. And it has constructions how to make these pistons of a certain size. Okay, but something, lightning or something hits it and changes something in the instructions in the computer. Okay, it's random, a random change. It then makes wider pistons. So just a random change happens to do something that's helpful. That's the theory of evolution. Okay. Not just in recent years, but it's becoming more evident in recent years. There are problems for Darwinian's theory of natural mutation, of random mutation and natural selection. And these problems are rapidly increasing, especially because of advance in studying DNA and other molecular biology things. And what I'm talking about now is not arguments between creationists and evolutionists. There's this growing conflict within science, within scientists, who are not necessarily creationists. They could be, but they aren't necessarily. Okay, problem number one. You probably heard of junk DNA. Okay, about 98% of human DNA has been called junk DNA. It's silent DNA. It doesn't seem to do anything. So you have two categories of you have genes that have instructions for making proteins. Protein coding DNA, it's called. The other part doesn't seem to do anything. It doesn't make proteins, and so it's been thought of as junk for years. Functionless remnants of evolution, stuff that used to be useful, but now it's no use, not useful anymore. Okay, are you willing to have 98% of your DNA removed? No, I don't think so. This junk DNA is needed by the evolution theory as a resource for making new genes. You could have mutations and they make this unneeded gene evolve into a new gene to make something new. Okay, so it's needed for evolution. But I remember in the early 1970s, my molecular biology colleagues at Loma Linda predicting that it would turn out that junk DNA is not junk. Because God wouldn't make something like that. Well, it's only the creationists who've been predicting that until recent years. There were some various papers began to appear which seemed to be saying, well, that silent DNA is doing something. It must be important. Like here's a graph. Somebody did a very voluminous literature study, and they determined that the percentage of DNA that's junk changes as the organisms get more complex. So you have prokaryotes, fungi, plants. These simple things don't have very much junk DNA. Who has the most? Humans. As animals get more complex, they have more junk DNA. Well, that looks suspiciously like the junk DNA is doing something important, like making them more complex. And I remember seeing through the years various articles coming out, like this one. This is one of my favorite. In the very prestigious journal, Nature. It's the junk that makes us human. So they're finding some of these junk DNA genes that are important for making us a human instead of some other animal. Well, papers like this kept coming out. Finally, it reached a climax here in 2012 with the completion of a massive government-supported research project, looking at all parts of the DNA and just trying to figure out what it's doing. And they published their results in 30 papers published at the same time. And they did that deliberately because they knew there's going to be kickback on what they were finding. Because they're finding that this junk DNA is not junk. It's in fact some of the most important DNA. Because it's regulatory DNA. And it controls what those protein coding DNA, genes do. So junk DNA is really no longer a useful concept. The supposedly junk DNA controls whether our protein coding DNA will make a human, a chimpanzee, or a mouse. You know, proteins like bricks. You can take the same bricks, you can make a doghouse or a palace, depending on what your engineering drawings tell you to do with it. And so proteins, you know, our proteins are a little different from mouse, but they're really surprisingly similar. And the junk DNA determines what to do with those other genes and what to make, a human or a chimpanzee. Okay, that's one problem, the demise of junk DNA. And evolutionary biologists generally are fighting this concept that there's no junk DNA. They don't like that. It's damaging to their theory. Okay, problem number 2. Evidence for the tree of life. This is the tree of life. The idea that everything comes ultimately from a common ancestor. All of our genes and all organisms evolved from a common ancestor. Okay, this idea is facing serious setbacks. And we don't have time to go into all of that, but we'll deal with a primary problem here. So this is supposedly the history of how all these changes happened. And remember, in Darwinian theory, all change has to be by random mutation and natural selection. And that includes all genes. Every gene that appears, every new gene that appears, has to have, according to Darwin's theory, an ancestry of some other gene gradually, little by little changing to make that new gene. A gene can't just pop into place. I mean, if that happens, it looks like somebody with intelligence is monkeying with the system. But what the ability to sequence genes is telling us now, that there are a lot of genes that are being called orphan genes because they don't have any ancestry. They don't have any ancestors. And then that's just not a rare thing. 10 to 20% of genes are orphan genes. There are no ancestors that they could come from. How do you deal with that in evolution? Humans have at least 1,000 of those. Orphan genes, and some are responsible for our large brain. And another example, the honeybee. Honeybees have this amazing ability to make honey. Okay, the genes that tell them how to make honey are orphan genes. Okay, so where'd they come from? They have no ancestors. Nothing like it in other insects. It looks suspiciously like somebody put them there because the honeybee is going to need it. And I think that's really is what happened. And so all these orphan genes are a serious problem for evolution. And you read the literature where once in a while an evolutionary biologist will try to explain this. And they go to interesting lengths to try to explain how this can be. One paper said, well, apparently, I mean, if you really are a committed believer in naturalism, in Darwinian evolution, there has to be an explanation for it. Sometimes you'll see papers that say, well, isn't it interesting how easy these things can happen? Even though they're impossible, if you really accept naturalism, you accept evolution, there has to be some way for them to happen, even though it looks impossible. And in fact, I would say it really is impossible. So orphan genes are a serious problem. And if you read what evolutionary biologists say, they're not dealing with this really well. Epigenetics. How many of you have heard of epigenetics? Okay, well, good. We got some. You've heard some things about it. Epigenetics is a fairly new field. Actually, it's been studied since the 80s, but it's becoming prominent in recent years. Epigenetics, it means kind of beyond the genes or outside of the genes, outside of DNA. I always wondered what this statement of the Bible means. Punishing the children for the sins of the fathers to the third and fourth generation. Well, now we know what it means. God actually knew something here. He wasn't just talking. Epigenetics is a management system outside of the DNA that determines how to use the DNA. Okay, there are sensors in the cell that detect environmental factors, and they put little tags on the DNA. I don't know if you can see them from back there, but there are little pink tags here, little pink molecules that put tags on the DNA, and that turns genes on or off, or determines how much they're going to work, like dimmer switches. Okay, this is a fairly new understanding. And in epigenetics is just blossoming, what they're finding, more and more things that are controlled by epigenetics.
per year. So we'll peg it at 1 centimeter per year, and we're going to plot this on a log scale. So 1, what's the log of 1? 0. There you go. So we plot that at 0. So that's the modern rate. That's the rate we actually see accumulating today. All right, let's compare that with radiometric dating. So we look at the rate of accumulation based on radiometric dates, and we get a rate very different from the modern rate. Even though geology claims the present's the key to the past, it's not true when you're looking at sedimentary rates, because if you look at radiometric dates, you find out that the accumulation rate is often about 1,000th of a centimeter per year. That's a centimeter. 1,000th of a centimeter would be like a single grain per year. Well, look at where that is on the scale. 10 to the minus 3 meters, way over there on the left. Very different from what we get in the modern environment. Well, then we have the Bible as a source of information. And in fact, the laboratory sedimentary behavior is very similar to what we see in the biblical rate, and very different from the radiometrically determined rate. Well, what do we do with all that information? Some people have tried to explain this. Geologists have recognized this problem, and some people have tried to explain this by saying, well, sediment's accumulating at the modern rate, and then it's all eroded away and more is accumulated, and then it's all eroded away and more is accumulated, and gradually, slowly, grain by grain, you build up a residual deposit. Well, if that were true, you'd certainly see evidence of it, because it can't always come back to the same spot again in the same place. So if this was the process that was going on, you certainly would see irregularities in the fossil record that would indicate this kind of process is going on. And in fact, occasionally you do find this, but the vast majority of sediments that we have studied show a continuous process of sedimentation without interruption, and those sediments are accumulating throughout the depositional process without erosion going on. All this being the case, don't you think it makes sense that maybe we ought to look at some other explanation for geologic time besides radiometric dates? Maybe it isn't millions of years. Maybe it's telling us something else. All right, number 2, sediments do not appear in equilibrium with the environment. What does that mean? It's too late in the day to talk in such abstract terms. So let's give it a little meat. This is where we're working in the Colorado Plateau, and we'll have to give you a lesson in sedimentology. Oh, where is it? There, bredimentology is what I call it. Okay, so this layer of bread represents a layer of sediments. I thank Dr. Brand for this idea. And so each layer is another layer of sediment. So here comes another layer on top of that, another layer, another layer, another layer, another layer. So you gather up these sediments layer after layer after layer. Now, how would you ever know if there was time between those two layers of bread? Suppose I laid one down, then waited a couple of weeks, and then brought in another one, put it on. What would be likely to happen? Well, yeah, you might get some bread mold. You might even get a lot of bread mold. And you might even attract a critter. So now, if you were to put the next layer on top of that, what would you see? Do you see the evidence for time? It's clear, and anybody can understand that. Well, in the case of the fossil record, those perturbations of the sediment aren't going to be mold. They're not going to be mice. But everywhere on Earth, there is bioturbation taking place. That's things that move into the sediment and move it around. If it's underwater on the ocean bottom, it's going to be burrowing organisms. If it's above ground, it's going to be roots and gophers and things like that. But sediment is never just going to stay the way it is for long periods of time. You can't lay down a layer of sediment, go off and leave it, and come back in 15 million years. How long is 15 million years? Anybody here know? Raise your hand. Okay, I'm probably about as close as anyone here to 15 million years. But I'm not very far ahead of you. So I have no idea what a million years is. I have no idea. And 15 million years is a long, long time. Can a layer of sediment, like a layer of bread, can it sit there for 15 million years and not change? Well, you know, I had a loaf of this bread, the one that I was using in this experiment. When I was done, it was cheap bread, so I just threw it on my shelf, and I forgot about it. And I came back in 6 months and revisited that loaf of bread, and you know what? It was in perfect shape. It hadn't changed at all because it was cheap bread and it had lots of cheese in it. So it was still unchanged, but that was 6 months. So I doubt that that would have gone on forever. So there are exceptions to that. Okay, so here's your pile of bread. Now, which layer has time in it? Bottom one, right. Okay, another indication of time is erosion. So if we have a layer of bread and it sits there for a while, you can have erosion taking place. And it's gone, and now the layers on top come along and they fill it in, and you can tell where there was time, right? Now, it may not be very much time. It doesn't take very long to erode sediment. But at least there's an indication that there was a disruption of the normal process. So we're going to look now at some sediments. This is the Moenkopi Formation in South Utah. And in these layers, you might not be able to detect it. But a sedimentologist could look at that and he could say, or she could say, there is no disruption of those sediments at all. They don't show any bioturbation. They don't show any indication of burrowing. It means they have been put into a milkshake blender and the button's been pushed. That's what happens. Destroys all the internal structures so that it would just be even all the way through. Because when it's deposited, these layers all show the characteristics of flow. And that may be fine structures like you saw in that picture. There may be some cases where it's very coarse. But there will be internal structures in there, and these can be destroyed very quickly by biological activity. And biological activity indicates time. How much time? Well, we don't know. But I'd like to see there's some time there, right? If this is deposited over 10 million years, I would expect to see a lot of these layers erased completely by the activity of burrowing organisms. There may or may not be. So, at these rates, all sediment deposited on a radiometrically determined time scale should be thoroughly homogenized, which means it's all erased. All the internal structures erased. There should be no boundary between layers. There should be no internal structure in the layers. It should all be gone because these organisms are going up and down like this, feeding on that sediment, and they wouldn't care anything about those internal layers. Instead, we find homogenized sediments are exceedingly rare. They're rare that you almost have to make a pilgrimage to a place to see them. Richard Bromley, who's an expert on this bioturbation, says 100% homogenization, 100% bioturbation of the substrate is the natural end product of the activity of the organisms that live inside the sediment. Failure to reach 100% or the failure of that state to be preserved in the rock record are conditions that require explanation. So if you see some rock, and by the way, next time you're out in the field, you look at the rocks and see if you see internal structure in them. If you see internal structure, then they didn't sit around very long before they were buried. And that means they were deposited rapidly. And if they're deposited rapidly, what do you do with all that 10 million years? Had significant amounts of bioturbation in them. And they're in layer 2. So there's a place where you might be able to put some time. But you can't put any time here or here. All right? This is another one. This is Combe Ridge. There's, I don't know, I can't read how many meters, 500 meters thick. That's a lot. 500 meters, that's 1,500 feet. And all of the sediment is in column 1. That means there's no time there. By no time, I mean very little time. Not enough time for significant bioturbation. Okay, let's jump ahead. Another one, column 1. Another one, column 1. All in column 1. Another one, all in column 1. The question marks down there mean that we couldn't see the sediments there, so we couldn't judge them. Another one, all in column 1. Another one, all in. Oh, look. This is what we're looking for. Right there. That's completely bioturbated. There's some little tiny spots that are completely bioturbated, but the vast majority is where? Column 1. And so these are exceptional. So in exceptional cases, we can find some evidence for bioturbation. But the vast majority of the sediments we look at show no indication of time. Another one, column 1, with just one little spot over here in column 4. Another one, column 1. Another one, column 1. Another one, column 1. That one, obviously, we couldn't see stuff in a lot of it. Another one's all column 1. So there we go through the whole geologic column in the Colorado Basin. The radar, there is a line, goes along like this, and there's 15 to 25 million years missing time right on that line. 15 to 25 million years. We studied this all across Colorado and Utah and Arizona. Well, some in Colorado, and Utah and Arizona. These are the places where we took pictures of the outcrops. Here's the problem. Here's the Moenkopi Formation down here. It ends at the lower Triassic, and the Shinnerup conglomerate is in the upper Triassic, and there's a long period of geologic time in there. So that would suggest that if this radiometric dating is giving us correct time, the Moenkopi sat there for 15 to 25 million years, or even 35 million years, before the Shinnerup was deposited. Okay? How long is 15 to 25 million years? And what can it do to the fossil record? Well, let me tell you how long it is. According to one very prominent geomorphologist in the Geological Society of America Bulletin, an article, he reports that in 15 million years, make that in 10 million years, that's even better. In 10 million years, all of North America would be eroded down to sea level at the rate of erosion that's going on today. All of North America will be eroded to sea level if the rate of erosion in the past is the same. That's what a prominent geomorphologist told us. The current rates of erosion extrapolated over North America would take all of North America down to sea level in 10 million years. That's how long 10 million years is. Okay, this is supposed to be 10 to 25 or 35 million years. There it is right there. This shows you that there's very little erosion there over long distances. This is 20 kilometers. And the top is eroded only a couple of meters difference in those two spots. Occasionally we find some erosion, like right here. It's down 2 or 3 meters there. It is real erosion, or maybe it's not. Maybe it's just pushing the layers down. We aren't sure yet. And here's what's even more interesting. At the surface where the two come together, we find load casts. Load casts are evidence that the material underneath was still soft when the material 15 million years later came in. So it still was mud cast, pushed down into this underlying mud and formed a cast of itself. Here you can see some other load casts up here at the top. Here's the contact that's supposed to represent 15 million years. Here's another deposit, another contact right there. There's a very thin layer of sediment right there. You can see it right here, which is made of a very clean white material. And it's all the way along there. It's not eroded. 15 million years it sat there without eroding. And yet it was still soft when the overlying material came in. In fact, look how soft it was. Here you can see that white stuff, that thin layer of porcelainite, pushed down in between the sediment that came in on top right here, and it just squished that down inside of itself. So it's still soft like mud. Here you can see the white porcelain here, and here is the sandstone that came in and pushed it up. So it squished it inside itself. Clearly, Moenkopi didn't sit there for 10 to 35 million years without erosion or deposition or lithification. That doesn't work. Something is wrong with radiometric dating. Erosional rates. At the current rates of erosion, California is moving toward Florida at about 1 foot per year. I've got some nice coastal property in California I want to sell you cheap. And I promise you in 15 million years, you'll be living in Florida and California. Because we can do experiments today on proteins and see how long they last. And we determine a rate of degradation of proteins that's appropriate. We can do this, we can speed this up by heating up the material under pressure and apply the rules of science, physics, and determine how long proteins will last. And at most, they last maybe a few 100,000 years under the most ideal conditions. Nobody expects a protein to last for millions of years. Nobody. So what do people say when they find these blood vessels and blood cells and proteins and even DNA in these fossils? What do scientists say? They say, whoa, that's really interesting. Or else they say, that's contamination. Nobody believes that it's actually there. But you can do the experiment yourself. You can take a piece of dinosaur bone. You can digest the bone away using a special solution. And you can see that there is still stretchy tissue inside it. You can do the experiment yourself. You don't need to be an expert to do the experiments. So how do you explain that? Something's wrong with radiometric dating. It's giving us times that are way outside the bounds of our expectations. Absence of bioturbation, rates of erosion, rates of deposition, and other features are not giving us correct values for real time. So I suggest that the explanation, the meaning of radiometric dates, it's time to reevaluate its meaning. Okay? That's a scientific explanation for something that I think is true because the Bible says so. And I don't need the answer to these experiments to know that God knows what he's talking about. Who am I to challenge God? What am I going to say in the judgment when I come before God's throne and he says, why didn't you believe me, my son? I told you what I had done. And I'm going to say, well, you know, radiometric dating told me the earth is millions of years old. And he's going to say, well, you know, that's why I told you what I did. That's why I told you, because I knew you couldn't figure it out by yourself. And what are you going to say? And I think it's very unwise to throw the Bible out. Years of experience, Dr. Brandon and I in our research, years of experience have taught us that God is, his word is reliable and it's a good guide for our thinking in science. Thank you. All right, let's bow our heads. Our Father, once again we approach you, the holy, mighty Creator of the universe.